Hi, my name is Sanne Casello, and today I'm going to be talking about stress and cognition through the lens of the kappa opioid receptor. Specifically, I'm going to be exploring the role of the kappa opioid receptor in cognitive dysfunction. I first want to introduce why researchers research opioid receptors. As you may know, there's been an opioid overdose epidemic in the United States. This graph here shows the increasing number of opioid overdose deaths in the past 20 years. This graph refers to any opioid, but when we're talking about opioids, we're referring to heroin, morphine, oxycodone, and fentanyl, all of which bind to opioid receptors. There are 4 opioid receptors: mu, delta, kappa, and nociceptin. We're going to be focusing on mu and kappa because of their role in emotion. Mu is associated with euphoria, or the euphoric sensation that accompanies a high after opioid administration. Kappa is associated with dysphoria, or the dysphoric state that accompanies withdrawal. This presentation will be mainly looking at kappa, or this dysphoric state. Now, what's interesting about kappa, or the kappa opioid receptor, is that it can be activated by stress. And so after a stressful stimuli, the neuron produces something called dynorphin, which is an endogenous, produced by the neuron, neuropeptide. Now, dynorphin is released into the synaptic cleft and then binds to the kappa opioid receptor, which shows the relationship between stress and kappa opioid receptor binding. This here is the dynorphin kappa opioid receptor system, the dynorphin core system. We're going to be looking at the dynorphin core system in the context of the prefrontal cortex. So you may have heard of the prefrontal cortex as the region of the brain that's involved in personality, higher level processing, and decision making. So we're looking at this region of the brain because we're interested in the cognitive dysfunction that occurs with opioid abuse. So we want to examine the region of the brain that's responsible for cognitive processing, which leads us to our 2 questions. 1, does morphine withdrawal release dynorphin to activate the kappa opioid receptor in this region of the brain, the prefrontal cortex? And if this does occur, where are the dynorphin neurons that cause this activation? To investigate the first question, we completed an experiment called precipitated morphine withdrawal. So precipitated morphine withdrawal is an experiment where a mouse is administered morphine for 5 days, 2 times a day. On the last day, the mouse is also given naloxone, which is a drug that you may have heard of as referred to as Narcan, this anti-overdose drug. So Narcan prevents opioid binding to the opioid receptor, so prevents any opioid action. When we give an animal naloxone, it causes the animal to immediately go into withdrawal. So we're able to evaluate and analyze this period of withdrawal. We take these brains of the animals that have undergone morphine precipitated withdrawal and do immunohistochemistry and microscopy to examine at the molecular level what's occurring. So we want to investigate, is there kappa opioid receptor activation in response to morphine precipitated withdrawal? So you may be wondering, how do you visualize the kappa opioid receptor activation? Well, to review, stress from this precipitated morphine withdrawal is going to cause dynorphin to be released. Dynorphin will bind to the kappa opioid receptor. Upon dynorphin binding, phosphorylation occurs. We can utilize antibodies, which you may have heard of in reference to COVID-19, where antibodies are able to target a specific protein region. And so we use antibodies that are programmed to target the phosphorylated kappa opioid receptor. So we're able to create antibodies that bind to this phosphorylated kappa opioid receptor, which would indicate dynorphin binding and activation of the receptor, and then apply another antibody that can bind to this antibody that only recognizes the site, and that antibody fluoresces. So we're able to visualize through fluorescence the activation of the kappa opioid receptor. So the results of this experiment show that when we give an animal morphine, but then give them saline or don't put them into precipitated morphine withdrawal, there's no kappa opioid receptor phosphorylation. However, when we do give an animal naloxone and there is precipitated morphine withdrawal, there's a significant increase in the number of phosphorylated kappa opioid receptors and a significant increase in the fluorescence of these receptors. We did a negative control using norBNI, which is a drug that blocks any dynorphin binding. So we showed that this effect is kappa opioid receptor specific. Here is a quantification of our results that show that precipitated morphine withdrawal caused a significant increase in the fluorescence due to phosphorylated kappa opioid receptors. Here is an accompanying graph where the behavior of the animal was quantified so that we could verify that the animal indeed was going through withdrawal. So to answer our first question, yes, precipitated morphine withdrawal causes dynorphin release that activates the kappa opioid receptor in the prefrontal cortex. Our second question is addressing where are these dynorphin neurons that cause this kappa opioid receptor activation. So in order to test this, we use something called chanorodopsin 2. Now, we injected chanorodopsin 2, which is a virus that causes there to be expression of a channel that activates a neuron, into p-dyncrete animals. So let's unpack this a little bit. As I said, chanorodopsin 2 is a channel that can be activated through blue light or laser light. So we can shine laser light using an optic fiber onto this virus, and it'll cause expression and activation of a channel that then can activate these neurons. Now, they're injected into p-dyncrete animals, which means that these channels are only going to be expressed on dynorphin neurons. So this technique allows us to examine dynorphin neuron activation. So we express chanorodopsin in dynorphin neurons and activate them. So we're activating just dynorphin neurons in a specific region, the prefrontal cortex of a mouse. Then we complete immunohistochemistry and microscopy to analyze whether or not the prefrontal cortex activation of dynorphin neurons leads to this phosphorylation of the kappa opioid receptor. Here are our results that show in green fluorescent protein, which indicates presence of chanorodopsin 2, and in red, CORP, or the phosphorylation of the kappa opioid receptor. This shows that when an animal is stimulated with chanorodopsin 2, so these dynorphin neurons are activated through blue laser light, that there's an increase in phosphorylated core. On the right, you can see that when the norBNI, a negative control, when norBNI is applied, then there isn't this phosphorylation, which indicates that this is kappa opioid receptor specific. On the far right, there's a graph that shows that there's a trend that there's an increase in the number of phosphorylated core in response to chanorodopsin 2. Ultimately, these results show that there are dynorphin neurons present in this region of the brain, the prefrontal cortex, that cause kappa opioid receptor activation. So, our conclusions. We know that morphine withdrawal causes core activation in the PFC, and we know that this is caused by dynorphin neurons that release dynorphin and activate kappa opioid receptors in the PFC. So ultimately, this indicates that local prefrontal cortex dynorphin release in response to morphine-precipitated withdrawal could be tied to cognitive dysfunction evoked by opioid abuse. Thank you.